Lessons for Biology. So hopefully you'll find this material interesting as well. Oh, that's not what I wanted to do. Okay, here we go. So in the biotechnology section, what I'm hoping is that you will be able to describe what biotechnology is, to describe various tools that we use in biotechnology, to understand how biotechnology has been applied to medicine and agriculture, and to be able to kind of just explain some of these tools that we use now for doing biotechnology. Of course, there's a lot more that I can't possibly cover. Biotechnology is a huge field, but if you find it interesting, we offer a master's degree in biotechnology. So it's a very popular field, very high paying jobs, and you make a big difference in people's lives. Okay, so when I say biotechnology, what I mean is literally the use of some sort of biological agent, whether or not it's a biological organism, a gene, a protein, something that came from or is a living organism for technological advancement. So here, what I'm showing you is the production of antibiotics. This is considered a biotechnology application. Antibiotics were first discovered because they are compounds, at least in the first antibiotic discoveries, they are compounds produced by fungi. And these compounds produced by fungi kill bacteria. That means we can treat our infections. So if you take microbiology or medical microbiology, we'll talk even more about the fact that we, the US, developed antibiotics first. I would argue really helped us win World War II, because we were able to treat infections in our soldiers, whereas the Germans and the Axis, they did not have the same type of treatments. So one of the first things that we often do in any sort of biotechnology application is we often will need to extract nucleic acids. Sometimes we extract them because we want to make copies. Sometimes we extract them just because we need to work with those nucleic acids. But being able to extract not only DNA, but also RNA from a cell is really critical. We are even using this to this day to diagnose COVID-19. The test to diagnose an active infection extracts RNA from your cells, turns it into DNA, and then amplifies it so that we can tell whether or not you've got the virus inside of your cells. So we need to be able to extract nucleic acids out of the cell without damage. In general, the process for extracting DNA involves lysing the host cell, usually using a detergent because that will disrupt that phospholipid bilayer. To destroy proteins, because we have lots of proteins that can destroy our nucleic acids. To spin, to centrifuge everything, to kind of get all of the thick cellular contents away from the nucleic acids. And then we can use an alcohol like ethanol or isopropanol to pull the nucleic acids out of the solution. This is a really fun experiment to do if you ever find yourself working with kids. You can extract DNA from strawberries. You take hopefully fresh strawberries, frozen are okay too. You mix them literally with like dish soap, water, and a little bit of salt. You smash up the strawberries. You filter out the chunks of strawberries, and then to the liquid you add some rubbing alcohol that you can buy at Walmart. I usually put it in the freezer first so it's nice and cold, and then you take a little piece of plastic and you can pull out all of the DNA. So it's a really fun experiment to do with kids so that they can see the DNA that's inside of cells, but you can extract DNA. And so the kids, sometimes the kids ask why, other times the kids are just so excited that it looks, honestly, it looks like boogers because it's like really mucusy and viscous, and so they They think that's cool because it looks like boogers. But sometimes the kids want to know, well, now why would you bother to extract DNA out of a cell? And they usually just say, why are you doing this? So why am I doing this? Well, one of the things that we may want to do when we extract DNA from a cell is to make copies of it. And right now, it's not important that you understand the details of how we make copies, because that's what you're going to talk about in one of your labs. So you will do a lab exercise on how to copy DNA. But the process by which we copy DNA is called polymerase chain reaction, or PCR. And the discovery of this process also won the Nobel Prize for medicine. So this was a really critical advance, because what it did was it allowed whoever, so law enforcement, clinicians, scientists, to start with very, very small amounts of DNA, and then to make millions and millions of copies of it so that they could do something else with it. So I like crime shows. That's kind of my guilty pleasure. In my work from home setup, I have a little TV like right above me, and I usually am playing like some old forensic files or cold case files or something. And if you watch old enough episodes, they talk about, well, you know, back in the 1980s when this murder was committed, you know, we only got a tiny drop of blood and we couldn't do anything with it because it wasn't enough. But now with, you know, 2020 technology, that little drop of blood is enough, because we can use things like PCR to amplify and make lots and lots of copies of that DNA so that we can do whatever it is we need to do to diagnose an infection, or to determine who murdered somebody, to identify of a crime. So PCR has been a really important avenue for improving lots of different fields in medicine, in science, and in law enforcement. One of the other things we do once we make copies is we sometimes run them out on a gel. I think we've talked a little bit about gel electrophoresis before. You will continue to talk about it in lab, but this is a way that we can actually visualize those amplified pieces of DNA. You see this person is wearing a shield. Usually what we do is we use a compound that is viewed under UV light. And so because you're shining a bunch of UV light, you have to protect your face and your hands, because it can burn you. One of the other things that we can do once we have amplified a piece of DNA is we can do what's called molecular cloning. So when I first learned about molecular cloning, I kind of confused it with actual cloning. So you have to remember, I'm old enough that I was either in 8th or 9th grade when they cloned the sheep, Dolly the sheep, in the late 1990s. So when I hear cloning, I'm like, ooh, they made a copy of a sheep. That isn't exactly what molecular cloning is. So molecular cloning is this idea that we can take a gene, any gene we want, we can make lots of copies of it, and we can actually put it into a bacterium, so the bacterium can make copies of the protein for us. And again, you might be wondering now, why would you want a bacterium to make a protein for you? Well, lots of reasons. Some pure research, but many medicinal applications. You may remember we watched that video earlier today of the student eating a donut, and then her body made all this insulin and helped her process the sugar. That's great. So insulin is a protein. Your body needs it to process sugar. And unfortunately, we have a number of people in the world who do not make sufficient insulin, diabetics. What do we do then? Because you have to have insulin or you're going to die. So we normally, prior to molecular cloning, would take insulin from animals like cattle, because they're big. But imagine how expensive it had to be to get cows to take their blood, to pull out their insulin, to purify it, and then to send it to somebody who's diabetic. Cost prohibitive. You can't afford it. Instead, we take the gene that will eventually produce insulin, we put it in a bacterium, and the bacterium makes insulin. I can grow bacteria forever. I can grow thousands and thousands and thousands of copies of bacteria in a cup this big overnight. In fact, I could grow billions of copies of bacteria in a cup that big overnight. And each one of them is making insulin. And instead, all I have to do is purify the insulin out of bacteria, and I have made more insulin than I could possibly get from a cow. I've made it more rapidly than I could get it from a cow. And I've made it far more cheaply than I could get it from a cow. I mean, I know we all live in the US, and so it's going to be expensive anyway, but in other parts of the world, it really has decreased the cost of insulin and made it far more accessible to people. So the idea that you can take a piece of DNA from one organism, like a human, and put it into a bacterium is called molecular cloning. Hold on, I'm going to close my window. Hey, buddy. My cat is terribly disappointed because he was looking out the window, but that's okay. So this idea of, again, taking a foreign piece of DNA, a human piece of DNA, they use this in cattle as well for cattle to have better milk production. They clone the gene for a hormone called bovine somatotropin, which is like a cow growth hormone. And they make bacteria make it, they purify this cow protein out of the bacteria and inject it back into the cow so the cow makes more milk. So there are applications for doing this in medicine and applications for doing this in agriculture as well. Now this is the type of cloning that I thought of when I first heard the term cloning. So we have cellular cloning and we have reproductive cloning. With cellular cloning, I'm really talking about the idea that unicellular organisms, when they replicate, the way they replicate is they basically make a copy of themselves. That is considered cellular cloning, when one cell makes a copy of itself. Reproductive cloning is what we all learned about, we being people my age and older, learned about when they cloned a sheep. Hey, Izzy. Sorry, the old lady walked. Izzy. Hey. Thank you. She's 12. She's really hard of hearing. And then when she hears things, she just goes off. So reproductive cloning is making a copy of an entire multicellular organism. And in theory, it sounds fairly easy. In practice, it was actually quite difficult, especially when we were talking about doing this with mammals. With things like amphibians, it wasn't quite as difficult. Amphibians are a little bit weird. They can do a little bit more like regeneration. Mammals are difficult. And so the way that this process worked when scientists were trying to clone a sheep was they had one type of sheep called a Scottish blackface. I don't really know much about sheep, but that's the kind of sheep it was. They took one of a regular cell from the sheep, and they did what's called enucleation. They basically removed the nucleus from this cell, and so instead it was just cytoplasm with all of the other cellular contents, but just no nucleus. Then they took another breed of sheep called a Finn Dorset sheep, and they took mammary cells and they basically electrocuted everything together and got the nucleus from the mammary cells into this enucleated cell from the Scottish blackface. that developed in the lab into a blastocyst, which is one of the embryonic stages of development. So they let it continue to divide and grow. They implanted it into a surrogate, and the surrogate happened to be the same type of Scottish blackface sheep. And then the surrogate gave birth to a sheep they named Dolly, and it was a Finn Dorset sheep because that's where the nucleus came from. So Dolly, the sheep, is genetically identical to the Finn Dorset donor of the mammary cells. So that's how you can clone a whole animal. Again, in theory, it sounds pretty easy. Take a cell, remove the nucleus. Take another cell, put the nucleus in that cell without a nucleus. Put it in an animal, let it incubate. Should work. It should work, but again, it is more difficult in practice than in theory. Reproductive cloning is one of those things that people are generally okay when you talk about doing it in animals. If you said something about doing it in people, you would get the side eye. This is not something you're allowed to do in people. But what you can do in people is what we call gene therapy. So there are a number of conditions that people can be born with that are a result of mutations to DNA, broken copies of a gene, and especially when we're talking about conditions that are caused by a single gene mutation. Again, in theory, the idea would be if you have a defective form of a gene, what if we could repair it or replace it? And then you would have a fully functional copy of the gene. So conditions like sickle cell anemia that are caused by a single gene mutation, or cystic fibrosis, which is caused by a single gene mutation. What if we could repair it or replace the defective or mutated copy with a fully functional copy? So that's what gene therapy would be, replacing or repairing a defective copy of a gene. The first methods that scientists have used to try to do this are using viruses. So the idea would be that you put the good copy of the gene into the virus. You allow the virus to infect the patient. And eventually, because of the way viruses work and the way viruses reproduce, that genetic material should get into the host cell. And because the host cell should recognize this as, hey, I have this gene, it would hopefully swap out the bad for the good. So again, in theory, it should work. In practice, sometimes it works, but there are going to be issues where viruses, for example, don't always bind to every kind of cell. So here they're showing you an adenovirus. Adenoviruses will bind to cells of the upper respiratory tract. They'll bind to cells of the conjunctiva, and sometimes they can cause GI issues. So they'll bind to cells of the gastrointestinal tract. Well, what if the mutation is in the blood? It's not going to work. What we have also seen in a handful of cases of this type of gene therapy is that patients mounted an overwhelming immune response to the viral infection, and that killed them. And there have been other situations where the virus integrated the gene, the new gene, into the wrong place, and the patients developed cancer. So again, in theory, it should work. In practice, it hasn't been quite as effective as we would have hoped. So then what do we do? Well, we still want to fix people. And so now we have this magic, I mean, I call it magic, it's science, but it's magical science. It's called CRISPR. The details of CRISPR are pretty detailed. They're not necessarily relevant for you to understand the class material. When you get into genetics and when you get into microbiology, we talk in a lot more detail about CRISPR. But the idea behind CRISPR is that it's a far more accurate and a far more targeted way to do gene therapy. Scientists have gotten very, very good at using CRISPR to remove bad genes. They are less good at replacing the bad gene with a functional version, but they're working on it. So there have been a few examples of using CRISPR in practice. One, in tomato plants, there's a mutation that leads to thin, like needle-like leaves. And when we talk about photosynthesis, we'll learn why that's bad. But basically, in order for plants to function, they need to acquire sunlight. They acquire sunlight using their leaves. So the leaves need to be as wide as possible for some of these plants because that's how they've evolved. They have to have wide leaves in order to do as much photosynthesis as possible. So in tomatoes, if the leaves are all narrow, they're not going to do enough photosynthesis and they're not going to grow nearly as well as they should. So scientists have been able to use CRISPR to repair that mutation so that a needle-like leaf will become a normal leaf. In cell culture, scientists have been able to remove HIV. So one of the really insidious things that HIV does is it integrates into the host chromosome. So scientists in cell culture have been able to remove HIV from the chromosome. And scientists are also working on trying to modify pigs in such a way that the pigs could then be used for organ transplant. So there are two reasons that we can't right now transplant pig organs into people. The first issue is that pig organs have immune proteins on their surface that our body says, ooh, you're from a pig, and it destroys those. So we'd have to be able to remove those. The second issue is that within the genome of every eukaryotic system, especially ones that have been evolving for a long time, we have remnants of retroviral infections. We have all this junk DNA. And sometimes that junk DNA actually isn't junk. It can do stuff. And we don't quite know exactly what it's doing. But when you mix the junk DNA from a pig with the junk DNA from a person, they all kind of get activated. And again, you start getting rejection of the organ. So scientists at Harvard have used CRISPR to remove all 62 of those pieces of junk DNA from pigs that cause us to reject their organs. So they have one breed or one line of pigs that lacks that junk DNA. They're working on making another line of pigs that lacks all of those immune markers. And you put the pigs together, and in theory, you have a pig whose organs could be placed into a person and there would be no rejection because the immune system wouldn't recognize it as foreign and those junk DNA pieces wouldn't become active and cause, again, rejection. And this headline was from last fall. There was an article where they've modified this CRISPR technique just a little bit. And again, in theory, this modified form of CRISPR could fix something like 88% of human genetic conditions. So that is obviously down the line, but that is what's kind of in the pipeline. Are pig organs almost identical to humans? Yeah. Especially in terms of like size and shape. Pig organs, again, in theory, are perfect for people. The size and shape wise, for adults. For children, I don't know, maybe you'd have to use a piglet, and then that, well, then there's a whole host of issues there too. But the idea would be, yeah, pig organs would be far preferable because people die on the organ transplant list every day, thousands of people. So nobody really developed CRISPR. CRISPR is actually a bacterial immune response to viral infection. So that's why we talk about it in microbiology. The discoverers of CRISPR were actually a group of scientists at Berkeley. Harvard gets a lot of press about it because Harvard filed the patent. Not first, they actually filed their patent second. Berkeley filed the patent first. Harvard filed second, but Harvard paid extra to file an expedited patent. So Harvard got the patent even though Berkeley figured it out and Berkeley was the one who was using it. So there's a big fight right now between Berkeley and Harvard with literally billions of dollars on the line and a Nobel Prize on the line. Whoever gets credit for this is going to get the Nobel Prize, I guarantee it. But there's a lot of fighting right now. So in my opinion, Berkeley should get it. Harvard was sneaky and they used all their like Harvard sneaky money to pay, but it was Berkeley. Yeah. And then of course, all of the scientists who worked on it at Berkeley were women and all of the ones who worked at it at Harvard, they were men. So I don't know, you can go however you want. But either way, as long as this technology is developed and is useful in humans, I think that would be a wonderful thing. And somebody is going to win the Nobel Prize. It just depends on who. So I told you we could use biotechnology to develop hormones. So this is showing you that example from cows where you take the cow gene for their hormone that helps them produce milk. You put it into bacteria like E. coli. You grow up billions and billions of E. coli. You purify out that hormone and you inject it back into the cow in order to increase milk yield. So when you buy milk and it says like hormone free, they're basically saying that this process did not happen to the cow. So they did not inject the hormone into the cow. So that's how you get like your organic, antibiotic free, hormone free. That's what they're talking about. So here are some human medical products that have been made by genetic engineering, by molecular cloning. So lots of blood proteins. So people who have anemia, people who have blood clotting issues like hemophilia, or people who have too many blood clots, lots of proteins there. Human hormones, wound healing, treatment of reproductive disorders. So especially in women who are having trouble conceiving, things like follicle stimulating hormone, insulin, of course, relaxin to facilitate childbirth, growth hormone to treat growth abnormalities, immune modulators as antiviral and anti-tumor agents, treatments for cystic fibrosis. So these are medical products that we now have that are accessible to patients for the most part because of biotechnology. Okay, this is another one of those where people get all like, ew, transgenic organisms, GMO. I'm not going to tell you whether or not they're good or bad. I'm just going to tell you what they are. A GMO is a genetically modified organism. Generally, we're talking about using biotechnology to directly add or change a gene product in an organism. So some people will argue, well, we've been genetically modifying organisms forever. Like, do you really think, you know, a banana looks like a banana? That's why bananas are going extinct, because they can't reproduce anymore. Do you think watermelon should have no seeds? No, watermelon are supposed to have seeds. So a seedless watermelon is a total genetic dead end. Like, we've been breeding, I have dachshunds. Do you think a dog is supposed to be like this tall and this long? No, right? We've been modifying organisms for millennia. Did we always know exactly what we were doing? No. Were we directly affecting specific genes? We didn't know it. You know, we were like, oh, we need this dog to be able to get into like a badger burrow and hunt a badger. That's what a dachshund is. It's a badger hunting dog. Oh, I need this big floofy dog to help me survive like in the Arctic. So you have huskies. Like, it's not that we, you know, were able to go in and turn on those genes we wanted. It was through selective breeding. But when we say a transgenic organism, or when we say a genetically modified organism, we are talking about directed changes to the genome of that organism, most often by adding a gene from a different organism. That's when we say transgenic. It carries genes from at least 2 different organisms, which is again not the same as crossbreeding or hybridization, We are talking about specific insertion of a particular gene into another organism. We use this a lot. For those of you who are not aware, almost every product that you buy in the United States that contains anything like corn or soy or cotton, like 90% of those products on the market contain genetically modified corn or soy or cotton. So just so you know. You can go to Whole Foods and pay a lot of extra money to get something that is GMO free. In order to modify plants, scientists have actually taken advantage of bacteria. So there's a bacterium called Agrobacterium tumefaciens. It causes tumors in plants. And it has a particular piece of DNA called a plasmid. So in bacteria, they'll have a chromosome, like a big piece of DNA that's their chromosome. And then sometimes they can have little tiny pieces of DNA called plasmids. And so this little tiny piece of DNA in this particular bacterium, in Agrobacterium, is what's responsible for causing the disease in plants, causing those plant tumors. If you are interested in plants at all, this is what causes crown gall disease, if you've heard of that, those big tumors that form on especially like oak trees and things like that. So what happens is that particular plasmid is able to get the DNA from the bacterium into the plant. And so what scientists have done is they've said, well, this bacteria is capable of putting DNA into a plant. What if we make the bacteria put the DNA we want into the plant? And so that's what they've done. So this is just a cartoon showing that they've modified this particular plasmid so it doesn't cause tumors in plants, but it still allows the foreign DNA to get into the plant. And then you do this in the plant cells as the plant is first growing. And if you know anything about plants, plants are super weird. There are lots of plants that you can just take like a branch and put it in the ground and eventually you'll get a brand new plant. So plants are super weird. They're fairly resilient. And so you use this bacterium to put the foreign DNA in, and now you can have plants that express your foreign DNA. So what are some applications of this? One of the first most major applications of this is making plants herbicide resistant. So herbicides are things like Roundup, like weed killers. So the main genetically modified crops in the US, again, soybeans, corn, cotton, and canola. So almost everything, like 90% is genetically modified. So herbicide resistance was engineered to protect crop plants from weed killers. So the idea would be, let's say you've got a crop of soybeans and you really want to grow your soybeans, but of course, if you've ever tried to grow anything, you know that weeds can come in and weeds will take water and they can choke out your plants. So you want to get rid of the weeds, but then if you spray something on them, why would it not kill your soybeans too? If it kills plants, it's going to kill everything. So a particular company you may have heard of called Monsanto engineered soybeans that are resistant to a particular herbicide called glyphosate, which they market as Roundup. So these are Roundup resistant soybeans. What this means for a farmer is that a farmer can plant these Roundup resistant soybeans from Monsanto in the field, spray as much Roundup as they want to kill all the weeds, and then they'll have all of their nice soybeans growing because they haven't been choked out by weeds. So this is what it looks like. These are regular soybean plants, non-genetically modified soybean plants. Here are the genetically modified soybean plants that have all been sprayed with Roundup. And so this is from Monsanto. So this is trademarked and owned by Monsanto. Oops. Another application is to make plants resistant to herbivory or insect consumption. So again, you plant your crop, you're so happy, and then here come the caterpillars and they eat your whole plant. Bummer, right? So scientists have been able to use that same system to put a toxin into the plant leaves. And again, that sounds scary. but it's a toxin that only kills insects. So it's a toxin called Bt toxin. It's toxic to moth, butterfly, beetle, and fly larvae, and mosquitoes. So if you do not have the toxin, you see the caterpillar ate the whole leaf. If you do have the toxin, the caterpillar did not eat the leaf. This toxin, again, is supposed to be harmless in humans. It does not affect mammals, but it does affect insect larvae. So these are options for farmers to grow crops to their maximum capacity, because they're not going to be choked out by weeds and they're not going to be eaten by insects, to improve crop yield, to improve profits, and to make sure that people have enough to eat. Because if you can improve crop yield and it can be at a lower price, people can eat. So, you know, pros and cons. It's whatever, you know, whatever your opinion is of a genetically modified organism, you at least know what they are. Scientists have also made genetically modified animals, so not just plants. The example I'm going to give you is fish. So many foreign genes have been expressed in research animals and in commercially important animals. So in this case, generally what they'll do is they'll take the foreign DNA and inject it into a fertilized egg. So then the entire organism, when it grows, would have that foreign piece of DNA. So there's a company, I don't remember what company it is, they make a salmon that's called Aqua Advantage. So apparently in aquaculture, for a salmon to get to like market size takes 3 years for them to get big enough to sell. These Aqua Advantage salmon grow in half the time. It takes them 1.5 years to get to market size. And so basically what they did is they replaced the normal growth hormone promoter. Remember, the promoter is where the RNA polymerase will bind for transcription. They replaced the normal salmon growth promoter, which allows the fish to grow in 3 years, with a growth promoter from a fish that grows much more quickly. I don't know which fish. It's probably proprietary. So now the fish produces more growth hormone because the promoter is different. It produces more growth hormone, it grows bigger faster. And so again, in the same amount of time, aquaculturists raising this type of salmon, the Aqua Advantage salmon, can raise 2 full crops compared to people raising conventional salmon or non-modified salmon. So again, these salmon, Aqua Bounty Technologies, that's a good name. These salmon are the same age. This is a conventional salmon. This is the genetically modified salmon. So again, your preference. Do you care if you eat a salmon that has a slightly different growth promoter? Or do you want to eat a salmon with a normal growth promoter? This salmon is, this big one right here, is probably going to be cheaper because the people raising these salmon are able to raise, again, twice the amount of fish in the same amount of time. These growers, aquaculturists right here, they are going to have to charge more because they are not able to sell as many fish in the same amount of time. So again, pros and cons. One of the other things we've been able to do with biotechnology, and something that is in the news right now, is we've also been able to make vaccines. So vaccines are compounds that when injected or ingested, when they get into your body, they elicit an immune response. The idea behind a vaccine is that you should be exposed to the vaccine. You will mount an immune response to that vaccine. And the next time you're actually exposed to the pathogen, like a bacterium or a virus, you won't get sick. So it's the way of tricking your immune system into thinking you're sick. It'll mount an immune response, but you're not actually at risk. And so scientists have been able to modify pathogens using genetic engineering. They can delete things from the pathogen that make you sick, but they can keep those things that would still stimulate your immune response. So honestly, I think vaccines and antibiotics are the two developments in the 20th century that most dramatically improved public health. If you go to like cities on the East Coast, we don't have a lot of them here, but if you go to cities on the East Coast and you go to some of the old cemeteries, you can see like a gravestone that lists like 5 or 6 names, and it's all the kids in the family, and they all died within about a week of each other, usually of something like diphtheria, for which we now have a vaccine. And so you would have families that had 11 or 12 children, but only 3 or 4 of them actually made it to adulthood because they would all die of these childhood infections. So now that we have vaccines, we've dramatically improved human lifespan. So we can modify pathogens to delete virulence factors, the things that make us sick, but to keep those that stimulate the immune response. We can take genes from a pathogen and put them into a harmless virus. So we're not at risk of developing infection, but we can still mount an immune response to that more dangerous virus. And we can have some vaccines that actually protect us against multiple diseases because we can put different pieces from different pathogens together. So I'm a big proponent of vaccines. I think they're very beneficial. I never got measles or mumps or rubella or diphtheria or whooping cough or tetanus. I did get chickenpox, but in my defense, the vaccine didn't come out until the mid-90s. I got chickenpox in the early 90s. It is what it is. I recommend them. And we also have these subunit vaccines that are only pieces of bacteria or pieces of a virus that, again, will help a patient mount a really strong immune response. So the idea is that genetic engineering and biotechnology have really improved agriculture, they've improved medicine, and so that use of biotechnology, that simple use of using biological systems for technological applications has been really important in improving the quality of life. So in terms of fewer diseases, but also in terms of access to better nutrition. Understanding DNA sequencing, transcription, and translation is really what led to these advances in biotechnology. So once we could sequence DNA, we could see, oh, okay, this is how insulin is made. Let's make a copy of this. Let's put it in a bacterium and let's make a lot of insulin for people. Biotechnology in agriculture has led to significant improvements in crops and livestock. So we now have herbicide-resistant plants, we have insect-resistant plants, we have cattle that produce higher levels of milk, we have fish that grow faster. So we have all of these improvements that can make food more nutritious and can make food more accessible and more affordable for people. And of course, there are lots of medical applications to biotechnology. We can replace missing gene products. So if you're diabetic, you don't make insulin anymore. Let's give you insulin. We can give it to you. You are a hemophiliac and you don't make clotting factors. We can give them to you. So we can replace your missing gene products. We can prevent and treat diseases through vaccines and through antibiotics. And we can potentially even repair defective genes. So again, biotechnology in medicine has dramatically improved the quality and the length of human life. Okay, so any questions on biotechnology? So when somebody tells you about GMOs, that's what they're talking about. So now you know. And you can decide for yourself whether or not you think they are good or bad. Okay, so I have 2 questions for you. So I'm going to open those up for you, put you in your rooms. Let's go ahead and go until, I'll give you till 10 o'clock, and we'll come back at 11 to finish up. So you can go ahead and join your rooms.